Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Anna Coussens. She's a laboratory head, part of the Division of Infectious Diseases and Immune Defense. She's part of the Walter and Eliza Hall Institute of Medical Research. Institute's very well known. It's been around for quite a long time. And she's talking to me from Australia. So, Anna, thanks for coming.
It's wonderful to be with you, Richard.
Yeah. Tell me about your research.
So my research focuses on tuberculosis, and I am an immunologist, which means I look at how the immune response controls an infection. And TB is one of the world's longest, or if not the world's longest running pandemic, but often forgotten about despite the large number of cases of each year and the huge amount of death that we see.
Yeah, what are the approximate numbers? How many people get sick with TB, and what percentage, what amount recover, what amount die? Yeah,
so a lot of our numbers around TB are collected through the World Health Organization. A lot of them are estimates because of the really poor reporting. So TB exists in sort of more lower middle-income countries as often the greatest cause of death in those countries. As I said, TB is often forgotten about in more developed countries, but it did in the beginning of the sort of 19th century really cause a huge amount of deaths as well. And throughout history, TB has caused the most amount of deaths for humankind. So every year, it's estimated we have about 10 million cases. So that's diagnosed symptomatic cases, and about 2 million deaths, which includes a quarter of those are HIV associated. So TB is also the number one killer of people infected with HIV as well. And so that kind of interaction, what we call a sort of a syndemic, these sort of two pathogens, is something that I also research.
What causes TB and what happens to a person that gets it?
Yeah, so TB is a bacterial infection. It's transmitted through aerosols, which means you can cough on somebody and they'll breathe in your cough and you'll transmit the bacteria that way. And so it starts off as a lung infection, and that's how a large number of people will develop disease within their lung. And you'll see, historically, you've seen people talk about consumption, or you see old movies of people coughing up blood into a handkerchief. That's generally TB because it causes destruction of the lungs. But it can also cause what we call disseminated disease. So it can move throughout your body, sort of like metastasis in cancer, and it can go to different organs. So we do see bone TB, we see meningitis TB, which is sort of in the lining of the brain. You get disease around the lining of the heart. And so and in people who have HIV, we do see a lot more of that kind of dissemination, where the kind of control is throughout the body is really not able to be controlled, I guess. And we see a lot of that in children as well. So children get really bad forms of TB, often in the brain as well. And it's much harder to diagnose in children.
What are the reservoirs of TB, though? Is it infected people, or are there animals or like certain living conditions that seem to be a reservoir for it?
Yeah, so super interesting. So, if you think about, there's a lot of contention about where TB originated from. As I said, it's sort of been with humankind for as long as we can have recorded history. We see it in, mummies from Egypt, etc. And we once thought that we perhaps got it from cows, but looking back at the genomic data, it now seems that perhaps we actually gave it to cows. Where we can also contract, and we did contract bovine TB back in the day, and pasteurization of milk was one of the things brought in to actually stop people getting TB through drinking infected cow's milk. So going back to where the reservoirs are, people who are infected, as I said, primarily get infected with these kind of places in the lungs. And so it's thought that it may sit in the lungs in what was historically called a dormant state. That kind of means the bacteria is not growing very much, and it kind of sits there and the immune system controls it. And that may be the case, or more likely we think that actually the bacteria is growing a bit and then the immune system's killing it a bit, and then there's a bit more of a controlled balance there. It may sit in lymph nodes. That's sort of one region that we think might be a reservoir, but it could actually be sitting in any type of tissue. Autopsy studies have shown that bacteria can be found sort of all over the body in people who didn't even get diagnosed with TB or die from TB. So kind of the harder you look, the more we find it. And that's something about TB, is that there are a lot of unanswered questions that in a way is inhibiting the field, because we don't understand really the extent of how the bacteria persists for such a really long time in people.
Well, what is the bacteria that causes TB, and are there different forms of it?
It's called Mycobacterium tuberculosis, so it's a Mycobacterium. One of the unique things about TB is that it's a really slow-growing bacteria, which makes it hard to study in the lab. So if you grow people who work on E. coli, maybe you've heard of E. coli, really fast-growing bacteria. TB takes 16 hours to double. So you have one bacteria, and then 16 hours later, you'll have two bacteria. So if you think about that, it takes us, say, 2 to 3 weeks to be able to kind of grow up a culture in the lab to work with it, or even see the bacteria, to be able to count it, which makes our experiments really, really long and really hard to work with. The bacteria has a really interesting cell wall, like an envelope, a really thick envelope around it, which makes it hard for drugs to get in, hard for us to be able to kill it. It's one of the unique things about the bacteria, and kind of what are the components of that cell wall of the bacteria, are unique ways as ways that it activates the immune system as well. We can get infected with some other types of TB. So there's more ancient strains, which are called Mycobacterium africanum, which exists in West Africa, and they form part of this TB complex, as we call it. So there are certain different types of sort of Mycobacterium that do cause TB-type disease.
When you say Mycobacterium, it's like fungi related. So why is it called Mycobacterium? What's special about it that gives it that fungi-seeming name?
Perhaps more it might be the lipid polysaccharide cell wall. You're right. So mycology could be more seen as a fungal infection, but this is just a Mycobacterium. Why it has a similar name, I've never really thought about it to be honest, but it's definitely a bacterium. So it might be that cell wall component, which is kind of a unique thing about it.
Well, okay. So studying this bacteria, it is culturable, although it's very slow, what you're saying. Has anyone looked at the metabolomics of it, the transcriptomics, the phages that prey upon it? There have been antibiotics created that affect it. I'm sure, I would hope that TB has been studied extensively. Yeah,
it really has. And people are developing phages, for example, as ways of targeting. We're using Bill Jacobs at Einstein is actually really developing these kind of unique fluorescent reporter phages that we can get into bacteria as a way of detecting, as well as using them as ways of, developing better kind of cellular models that we can actually detect the bacterium early, for example. As I said, it's difficult to study because it does take a long time to grow up. So lots of people work on the metabolism of the bacteria and trying to understand new drug targets, for example. And as I said, one of the reasons why it's difficult to often treat is because of this slow-growing sort of metabolism of the bacteria, which means normal drug targets, which target replicating bacteria, don't work very well when you have these somewhat dormant, non-replicating bacteria. So that's one of the difficulties in treating it. And perhaps treating earlier stages where if the bacteria is not growing, most of our drugs don't work.
Does it have a stage where it goes into like a spore formulation, or does it have a life cycle? Like, does it form biofilms? What else is known about it?
Yeah, no, it doesn't form spores. It does have interesting secretion systems. So it's not necessarily thought to secrete toxins, for example. So, but it does secrete virulence factors, which will activate the immune system. So the EX6, CFP10 through the EX61 secretion system induces interferon gamma response within the host. And actually, if you knock out that locus, it does become an avirulent bacteria. And BCG, which is the kind of bovine form of TB, which was developed into the only TB vaccine, is actually missing that locus. So we do know that these secretion systems are really important for the bacteria pathogenicity. There's a lot about the bacteria we don't understand. There's a very large, what are called PP repeat regions within the genome, which they're highly repetitive regions, and they're thought to potentially contain virulence factors, but conventional sequencing technology was really poor at getting accurate sequences because of that repeat region and the slippage. And so using nanopore technology or sort of PacBio, we're getting more long-read sequencing technologies to be able to look within these regions and see what they may be contributing to the pathogenicity of the bacteria. So that's all really recent research that's been coming out over the last few years. So as our technologies are developing, that we're learning a lot more and trying to sort of treat the bacteria differently.
Well, okay, so what is it doing in the various body parts? What does it look like if samples are taken from cadavers or from live people? The bacteria just proliferates, and what does it do? Does it do specific things in different body tissues? Does it interact with various tissues in different ways?
Yeah, so I guess that's, for me, that's the interesting thing, right? So I work on the host response to the bacteria. Typically, you think about treating a bacterial infection, you develop drugs to kill the bacteria, and that's how you resolve it. The thing about TB is that most people who get infected don't develop disease. And as I said, TB is such a historic bacteria and kind of human pathogen that essentially we co-evolved. And you can see that if you trace the evolution of the bacteria, we see different, clades, what we call sort of ancient and modern strains, and there are different lineages of TB. And we see that they actually correspond to human migration patterns out of Africa and how then populations grew in either sort of Asia or North America, etc. And then we see that colonization back into Africa actually brought these different strains then back in. So we think that there's a lot of, it could be even symbiosis in a way, that the bacteria and the host have sort of co-evolved, right? Which means that the majority of people have a very good immune system that they can control it and not develop disease. So my work is focusing on trying to understand, well, in the people who do develop disease, what goes wrong? And can we understand actually where this protective immune response has led to a detrimental response, which leads to disease? So as I said, the bacteria doesn't release toxins. It doesn't degrade the tissue in any way that, causes pathology. The pathology is caused by an exacerbated immune response to the bacteria. And so it's an immune pathology, which generally destroys the lung by the host factors destroying the lung matrix. And that's one of the things I work on, is trying to understand how host cell death, immune cell death, which is one form of how the host cell tries to kill the bacteria, actually leads to breakdown of organ function and tissue function.
Well, what happens specifically? What does the host response look like? What does it mean? So
primarily we think that, macrophages as phagocytes are the first cells which take up the bacteria. So I work on macrophages, and also neutrophils is sort of the other major phagocytic cell. Neutrophils have, they're very short-living cells, and they kind of either die from apoptosis, which could be a protective response, or they can die from a necrotic form of cell death called neutrophil extracellular traps, which is when they essentially unwind their DNA and they have decondensed chromatin, and this DNA mixes with the proteins within the granules of the neutrophil, and then they're released in a web-like fashion as the cell dies. And that's done to trap extracellular bacteria and try to kind of mop everything up, right? But because the proteins within the nets have a lot of enzymes which are involved in matrix destruction, that's there to allow cellular infiltration to assist in combating that infection that's there. But when you have a continual cycle of that and non-resolving inflammation, that's when you end up destroying the tissue around it. So essentially, my work is trying to understand what controls the balance between an acute response, which is beneficial, to then what turns into chronic inflammation and a chronically bad response, which then, destroys the tissue.
Well, all right. So, I mean, has anyone tried to develop antibiotics against this bacteria, or like what has been tried so far, and what's happened? What's unique and different about this?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
We do have antibiotics. They came through to the 40s, the 50s, and that's how we sort of first got on top of the TB epidemic. It's been a really slow developmental pipeline, I guess, since then. As more developed countries, the epidemic came under control, TB began to be forgotten about in a way. And when HIV came along, particularly that then lead to those individuals, as I said, were at increased risk of developing TB, and we started seeing the epidemic take off again. And that's where more focus came back on TB. So there was a number of probably decades where there was little investment in TB research. And only the last couple of decades has there's been more sort of money put back into it again. And so new drug classes are trying to target, as I said, different metabolic states of the bacteria. And there are some new drugs which are coming out. Essentially, one of the ways we tried to deal with the epidemic was instigate what is called directly observed therapy. And this came about because poor treatment regimes were being used, and that was leading to a lot of drug resistance emerging because of the long time requirement of treatment. So we generally need 6 months of therapy. And so DOTS, which is this directly observed therapy, was initiated by the WHO, which essentially means that every day you come to the clinic and someone observes you taking your drugs for this 6-month period. So you can imagine what that does to an individual, their ability to have a normal life when every day they have to go to the hospital just to have their meds because someone has to observe them because no one trusts them to take their meds, right? Which means that we have a very singular approach to treatment now. It was a way of getting on top of the epidemic, but now everybody gets treated in exactly the same way until we determine that you have a drug-resistant infection, and then you actually need to go on generally therapy for 2 years. And still there's only really a 50% success rate in treatment for drug-resistant TB.
What does the immune response look like to TB? Has that been observed? There are macrophages that try to engulf the bacteria, or, do they release substances to lyse the bacteria? Like, what's the response look like normally?
So a good response would be the macrophage phagocytoses it, puts it into phagosome. Phagosome gets, lysosome fusion, and then you get degradation of the bacteria and thus control of the infection. That's what should happen, with any normal bacteria. As I said, this ESX1 secretion system with the ESX6 and CFP10, they put pores into the phagosome membrane and actually then activate the gamma system. And by degrading, sort of putting pores in the membrane, the first thing that the bacteria does is try to escape the phagosome. It arrests phagolysosome maturation, so you don't get that degradation. And that's the first way the bacteria sort of begins to survive within the macrophage.
Wait, so it's after it's been engulfed, it tries to poke holes and it's like a whale swallowing someone, and the person's trying to rip out through their stomach.
Yeah. Yes. Nice analogy. Yep, exactly. So it's exactly punching through the phagosome, and it can escape into the cytosol. It does some very interesting things. So, as I said, part of these virulence factors which it secretes, ESA6 CFP10, it's been shown that it can actually shuttle those for antigen presentation and essentially release vesicles which contain that. Other phagocytes will then actually take those up, and then you'll get sort of, presentation of non-infected cells who are presenting antigen. And it's thought that, and the infected cell will not present the antigen, but release the virulence factors through these vesicles.
The bacteria is causing the phagocyte to release customized extracellular vesicles that are signaling other macrophages to do what?
Who then, I guess, take up those vesicles, and then they present antigen. So you can then imagine that T cells coming in recognize non-infected cells who are presenting processed antigen, and they're not recognizing infected cells because the bacteria is inhibiting that cell presenting.
Oh, it's inhibiting the presentation of antigen.
Yes, the infected cell would be inhibiting, but releasing these vesicles, which are then taken up by macrophages in the environment. And then they take up those vesicles and then actually present those antigens. So you get influx of T cells, but the T cells then are recognizing the non-infected cell versus the infected cell.
What's confusing the T cells so that they don't recognize infected cells? Like, what's changed about it sounds like the vesicles are doing it, but what is changing about the T cell itself where it doesn't recognize, it's still alive, but why doesn't it recognize infected cells now? Or bacteria,
that one study I was explaining sort of showed that it's stopping the infected macrophage presenting the antigen, despite the fact that it's kind of releasing the antigen via these vesicles, right? So it's releasing via vesicles, but stopping the normal transport process to presentation.
So, okay, I see what you mean. So it's infecting, the macrophages are doing their job, eating the bacteria. The bacteria, it's like that picture of I think it was a stork eating a frog, and the frog's arms have come out of the throat to like strangle the stork to stop it. But all right, so I see what you mean. So they're making the phagocytes, they're infecting them, they're stopping them from consuming them, and they're also making it look like they're not infected as well, the phagocytes. So maybe that signals the rest of the body not to produce as many as it normally would, because it thinks they're on the scene and working.
Yeah, or it's kind of come in but actually target another cell, and maybe you want to kill that cell, so you kind of kill the good cells, the non-infected cells, and then you don't kill the infected cells. It's kind of the way that the bacteria kind of subverts the immune response for its benefit to survive and kind of probably cause damage to other cells. So I guess if you think about it, what the bacteria requires for its life cycle to perpetuate is transmission, right? And for transmission to occur, it requires lung matrix breakdown, because that essentially it needs to escape the macrophage, get into the tissue, extracellular space, and have the tissue breakdown such that it can actually get into an airway and cause enough damage that you cough potentially, and then can transmit, right? So the bacteria requires the host to essentially degrade itself and have a bad response to perpetuate. If most people, perhaps the good response essentially is macrophage phagocytose, they present, they recruit T cells, we get more cells coming in, and essentially you form what's called a granuloma, which is a complex of macrophages, neutrophils, T cells, B cells, etc., which form like a ball, classically a ball, in which walls off the bacteria from the rest of the body to prevent escape. And we see that you do see granuloma classically within lungs, and you can see what's called the Gone focus, which is what we thought is the sort of primary side of infection in the lower lungs. And you see that, you can even see that on sort of CT scans where you have a really dense, sort of even a calcified nodule, and you get like a little discrete nodule in the lungs, which could be this controlled infection site. And that might be how many people respond. It's when that controlled response breaks down, and the ability for a really balanced immune response to control that granuloma and not allow the bacteria to escape and not allow the bacteria to cause cell death. Because if you have more cell death, then you lose the control of that little structure.
Well, is the goal of the bacteria just to proliferate, or is it to infect cells more and be more virus-like? I mean, like when it infects a macrophage, does it keep the macrophage alive for a long time? Like what happens to it? What's the fate of it?
It does replicate quite a lot. You can see, as I said, it's a slow replication, so you may get doubling in a day, etc. Right. But there is a stage where you will end up with, if it continues to replicate, if it escapes into the cytosol, for example, and replicates, then yes, you will end up with different forms of potential cell death. I work on sort of pyroptosis, necroptosis, these kind of inflammatory forms of cell death, to see how the bacteria triggers that. What we have found is that different clinical strains of TB can elicit essentially different forms of even cell death, or different degrees of cell death, I would say. And one of the things that my work is trying to look at is some people have really bad cavities, so really bad TB, where the cavities is when there's a really large destruction of the lung matrix. Some people present with TB and don't really have these large cavities, but the kind of cavitary lesions are associated with really high bacterial load. The bacteria actually does seem to then grow in that kind of airspace within the cavity in the lung, and you see, in a way, do get like a biofilm when the bacteria really loves to grow there. And that's a really great way for it then to transmit sort of high bacterial burden.
So that's like niche construction. But do you know what happens inside the again, the macrophages that are infected? Do the bacteria have a long lifetime in there? Like what do they tend to come at? Do they hide from the immune system and then they kill the macrophage and eat it and use it as material and then grow, or like what happens to them?
Yeah, so they do use the material and grow. So we have seen that in our cultures, if we even lyse the macrophages, you'll get a lot more extracellular growth of the bacteria compared to having bacteria in a well without these kind of lysed macrophages. So they certainly use the macrophage carbon as a carbon source for increased growth. I think that could be one reason why they actually like to kill the cells and then live in that extracellular space, because they feed off of it. They often take the cholesterol from the macrophage. So your question though around, do they like to sit and replicate or do they kill? The reason why I can't give a good answer to that is because each cell actually seems to respond differently. It's not a homogeneous response, and this is one of the most complicated things with TB. We do a lot of
each cell type.
I think my work focuses on, we very much use human cells. We always make human primary macrophages. But when you do that, you'll have heterogeneity even within a culture, right? And so this kind of subpopulation level heterogeneity means that each macrophage may respond differently. And it may be determined by perhaps how many bacteria it initially encounters. Certainly if you whack a culture with, MOI of around 10, you're going to kill all these macrophages and they're going to go into necrosis really quickly. If you do low MOI, which is what we do, to try and see what happens if you may infect 10% of the culture and then monitor that over time, even after 7 days, you'll get a lot of cell death still. But that cell death is increased if you take different clinical strains with different cell wall phenotypes. So there's an interaction between the host and the bacteria, which in some certain combinations is really bad. And as I said, so some people can control and some people don't control very well. And my work is trying to focus on what are the different sort of subpopulation responses, and we're looking at single cell level to really try and tease that apart. What is a state where the bacteria doesn't grow well versus does grow well?
In an advanced TB infection, what do you see? Do you see a lot of TB bacteria everywhere, or do you see relatively few ones, and is it surprising? And instead you see like a lot more niche construction and cell death. Like, what appears to be the end game of TB within a given tissue? What does it tend to do?
Yeah, I guess the end game certainly is, if you look in autopsy specimens, for example, or just even biopsies, you will see extracellular bacteria either in the caseum, which is kind of the liquefied necrotic center of a lesion. Either a cavity is full of caseum, it's just dead cell debris, which is where the bacteria can often be. You will also see the bacteria within cells still, so it'll exist in both states, but certainly it needs to escape into the extracellular matrix to get to a disease state, because essentially when we're diagnosing people, it's like end stage. And my work, we have been using sort of high resolution PET-CT imaging as a way to look at early infection in recent TB contacts. And when we do that only really detects inflammation using the PET response, which is a measure of essentially, it's a radioactive glucose analog. So it measures metabolically active cells, which are primarily the kind of the neutrophils and macrophages responding to that infection. And we can see really strong responses and inflammation on the lung in people that last for years before we can sort of detect the bacteria in their cough, in their sputum. But it doesn't mean it wasn't there before. It just may have been controlled perhaps in that within the cellular milieu and not actually, I guess, degraded the matrix enough that allows the bacteria to escape.
Well, you said when you're able to see it, it's usually quite advanced to even diagnose it. But again, if you look at, let's say, a deceased person recently from TB, what does their tissue look like? Is it just a massive abundance of TB bacteria, or is it more fewer ones that are in biofilms and there's like niche construction going on? Like, if you were to guess and look forensically at a whole bunch of people that have died from TB, looking at the tissue, like forensically, what does it tell you? What do you see? Is there anything unusual you see, or what do you see?
What you see is that the normal architecture of the organ is destroyed. So you'll have massive immune infiltrates. So a lung should be a gaseous sac. It's full of alveoli, but what you have conversely is just massive immune infiltrates, so that the lung matrix is just wiped out. And then within those areas of infiltration, you've got areas where what we call is this caseous necrosis. So you'll end up with, it's like a liquefied, it can be some of the times it can be calcified, and sometimes it's liquefied, which is just cell debris. And the bacteria, we do see a lot of bacteria in that area. It's actually thought that part of the difficulty in treating TB is that the bacteria that's within that caseum, so really high bacterial numbers, it's very hard for the drugs to get into because there's not enough vasculature to get the drugs in. And the bacteria in that kind of sort of it's even a kind of a fatty state in a way, is also could also be slow growing and doesn't respond to the different drugs. And certainly it's been shown using certain imaging techniques that we can see which drugs get into that area or not. And some drugs can't even penetrate that part of the lesion. But there are a lot of bacteria there. But you will also get some people who don't have that much bacteria. People do present differently. But
when they do, what's different? So some have a lot of bacteria, some don't.
Yeah.
But what else is very different about, what you see? Are you surprised that some people have died at all? Does it not make sense, or does it always make sense because the tissue is just so modified and destroyed, whether or not there's a lot of bacteria in it?
Often, I guess, perhaps the reason for dying is when obviously your lungs do, I guess, if you don't have enough oxygen exchange, you're going to die, right? So that's part of, I guess, one of the causes of deaths will be that your lungs are no longer functionable. I guess the interesting thing about TB is you'll have some people, and actually quite a lot of people may have a discrete lesion cavity in the right upper lobe. We often see that you can have one part of a lung infected with disease and another whole part of a lung with no apparent abnormality. So some people present with unilateral disease, while some people present with extensive bilateral disease, extensive cavitation, etc. So that's where people present differently. And as I said, some people also develop spinal TB or meningitis TB, where the bacteria can escape and lead to disease in those areas. And so again, that's where in the spine, you'll get a breakdown of the bone matrix and you'll end up with deformities. And so those kind of end stages really causing destruction to organ function because the tissue structure itself is degraded.
Are you able to tell that the degradation is because of the host immune response, or could there be other reasons why there's such tissue degradation?
That is something that we're looking at. So we've looked at sort of MMPs for a long time as these matrix metalloproteinases as enzymes which degrades collagen, elastase, etc., which is the kind of the structure of the tissue. And that is sort of part of the work we're doing, looking at the nets and how the nets will degrade that. I'm actually starting a project now to look at spinal TB, to actually look at the pathology within those lesions to learn more about what's causing that matrix breakdown. As I said, one of the interesting things about lesions sometimes is that they do become calcified, even within the lung. So you have these sort of calcified nodules. And so there's a question around how bacteria may even modulate sort of immune cell programming to become, I guess, more kind of even bone-like, right? If you have stem cells within the lung, myeloid cells and bone cells are sort of the same lineage, right? So you could potentially even think about sort of reprogramming stem cells into becoming more sort of even osteoblastic cells, perhaps. And so maybe the bacteria is modifying even that bone process and ossification. So these are kind of questions we really don't understand, but we're beginning to think about them. There's some recent research showing that the bacteria can also exist within stem cells and modify the stem cell differentiation program such that you get different phenotypes of myeloid cells coming out from the bone marrow itself. But that's sort of in some animal models.
So, I don't know, do you feel like you're close to any breakthrough in understanding of the immune system in TB, or is it just, I mean, like, what hypotheses do you have currently? Like, what are you going to be testing over the next year or so to figure things out?
Yeah, so I think we have. I mean, I sort of tried to, I guess I've described a lot of complexity, but I said my research really tries to understand individual response and the interaction with the bacteria. And we have been identifying a number of host cell death pathways, which we are identifying sort of small molecule inhibitors to prevent that cell death and actually lead to a better resolution of the infection. So we will like to try and put those into sort of host-directed therapy trials. And so my work is trying to think about not just treating the bacteria with an antibiotic, but actually trying to treat the individual and potentially the kind of immune dysfunction of that individual, which is leading to disease. And so we effectively want to pair antibiotic therapy with host-directed therapy with the idea that we want to resolve the inflammation and lead to better outcomes of treatment. Often if we, what we're seeing is that, people are treated with antibiotics as this kind of baseline therapy. And we do recognize there is essentially what's called sort of post-TB sequelae, because people do end up having poor lung function afterwards. TB treatment is not a cure, because people who have TB are at the highest risk of getting TB again, if infected again. And that's sort of some of the poor language that's used when people are told that they're going to receive therapy is often they think that they're kind of cured and won't get it again, but actually they're at high risk of getting it again. And I think a lot more work needs to go into, what therapy can we give those people to actually, improve the immune response such that on a subsequent exposure, they don't get TB again. Because that's the, I guess, one of the big perpetuators of the disease in high burden settings. Okay,
well, very good. What's the best way for people to find out more about your work? Where can they go?
Cool. They can find me at Twitter at Anna Coussens, and also on the WeHi website. You can look me up there.
Okay, well, very good. Well, Anna, thank you for coming on the podcast. I appreciate it.
No worries. Thanks so much, Richard.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.